Hello, this is Nuria Montserrat, Barcelona. First of all, I would like to thank Nature and Nikon for this kind invitation for us to present part of our work for the creation of kidney organoids from embryonic and pluripotent stem cells as an infection model for SARS human pluripotent cells, including both embryonic stem cells derived from human embryos and induced pluripotent stem cells, have the inherent capability to give rise in vitro and in vivo to cells of the three layers of the embryo. This capability has been exploited in different laboratories, including our laboratory, to try to understand how human tissues develop with the final aim to perform for tissue and human disease modeling. All this knowledge in the lab has also been key for the development of three-dimensional organ-like organs that are called organoids. When derived from human pluripotent stem cells, including human embryonic stem cells or induced pluripotent stem cells, contain multiple organ cell types. These organoids are shown to be capable to recapitulate some specific organ functions, and very is that in organoids we can also see that cells are as fashioned as in the final organ. In my laboratory, we are particularly interested in the derivation of human pluripotent stem cells. But first, I just want to use these schematics from Melissa Little's work, in where you can develop in the mice. In magenta here, you can see this small piece of primitive streak that indeed during embryo development is going to give rise to the intermediate mesoderm. Intermediate mesoderm is the main tissue from which, and in this cartoon you can see that the posterior part of the intermediate mesoderm in yellow is going to give rise to all the tissues in the kidney. The green part of the intermediate mesoderm, which is called anterior intermediate mesoderm, may be during development to progenitors, which are the progenitor cells of the mature kidney. These steps have been widely in the mice. By studying these steps in the petri dish, our objective some years ago was to try to derivate for the first time that were characteristics in terms of gene expression, as we can see here by qPCR analysis, and also by immunofluorescence. Only after 4 days of the exposure of into different conditions, we could derivate for the first time ureteric buttons. But in order to find the differentiation potential of these cells, we took a system called reaggregation assay, in where we further aggregate our ureteric bat cells together with embryonic kidney cells from mice. This assay has been undertaken by states, and as you see here, when one takes associate embryonic kidneys from mice and we put them back in these organotypical conditions, the cells are going to be reaggregated, and during 4 to 6 days, we can follow will develop. When we reaggregated our undifferentiated pluripotent stem cells together with embryonic kidney cells, we can observe already in this able to derive any rudimentary structure like in this mouse control assay. On the contrary, when we 4-day ureteric progenitor cells derived from human embryonic, we could observe that these cells were able to execute and to induce this tubulogenic potential. So we also label the cells with a lentiviral vector for the expression of also GFP position, and we can see that ex vivo cells are localized in the of the structure in the organotypic culture. So this was the first time, and this study was run when I was a postdoctoral fellow on the epithelial laboratory. This was the first time that we could show to derivate human pluripotent stem cells related by progenitors that were capable to give rise to this chimeric kidney. Or laboratories, as you are probably aware of, have been also showing the potential of human cells to give rise kidney organoids. These different signaling pathways by using different cogs and growth factors that are mimicking important steps of kidney development in the petri dish. So we observe that there are compared these different protocols in terms of the time length that this protocol also with regards to the differentiation potential of this kidney. However, the field in the last 5 years has truly advanced, and we can observe kidney organoids have been already amenable for testing nephrotoxic agents, also to understand the features of disease and treatment. And also we want to highlight that in 2015, the organoids derived laboratory were transcriptomically named as first trimester gestational kidneys. Of all these important findings, we decided to propose a protocol in our further derived kidney organoids from human pluripotent stem cells by mirroring important steps of kidney. So we first induce the formation of primitive streak from progenitor cells, they derive kidney organoids. And our hypothesis was that if we were going to assemble and to aggregate intermediate progenitor cells derived from pluripotent stem cells sooner than the other, forcing cell-to cellular matrix contact, probably providing kidney organoids in a shorter time and with higher characteristics compared to other findings. So in a first approach, with streak cells, that as you can see here, were which is a pluripotent stem cell marker, while we could observe the detection of brachyury also by FACS analysis. And also by qPCR, we could observe that the phenotype of the cells was of the primitive streak. Then we further exposed the cells into FGF9, trying to mirror as well important signals that the cell in development, and we observed that only by exposure by one single day, cells were carrying the expression of FACS2, which is an important marker of faith by presence and also by FACS analysis. And we also the cells by using qPCR analysis. Then, in order to further potential of our progenitor cells, we took again advantage of the reaggregation assay by assembling day 5 nephron progenitor cells together with mice from kidneys. And we can see here in red are stained for this human nuclear antibody. Hello, that were grafted from laboratory in green, and Nikon for this kind of invitation for us to in the part of our work here in life. Of kidney organoids from embryonic and pluripotent stem cells and an infection model human cells were found in different structures in these reaggregates. And these results were further confirming the different progenitor cells. So we further proceed and culture these progenitor cells in transwell system, and by 16 days of diffusion, we could obtain these kidney organoids that were also RNA-seq, and we compare our data together with already, and we that our kidney organoids already at this situation were transcriptomically similar to second trimester gestational kidney. Also, our shown that when we took advantage of confocal analysis, that our organoids were like structures that were segmented and expressing important proximal tubuli and also important markers of the glomerular compartment. Of course, the extent, differentiation, and localization of all these markers together with human fetal kidneys of 22 weeks of gestation. Importantly, we were also finding endothelial cells scattered structures, but these were not as in the native kidneys. So, in order to further solve this question and to explore a new providing a vascular environment into our key organoids, we made use of the cheek chorianthonic membrane assay. The cheek chorianthonic membrane assay is a high vascularized extraembryonic tissue that has been extensively used for tumor angiogenesis research and also for grafting biomaterials. So more vivo models, such as the mouse, they can represent natural immunodeficient environment that can offer direct and minimal invasive access to the assay site. Our organoids from day 7 of development, and then track the samples at day 12 of this time course. And as you can see here, in this next slide, we took advantage of the injection of dextran in ovo in order to further organoids were properly grafted into the video shows here the reconstruction of these confocal images. We can see here invading our kidney organoids. Of course, we took advantage of more sophisticated assays in order to put kidney properly grafted into the cheek and also differentiation extent of our kidney after implantation. And as you can see here, consecutive sections of experiments, and by hematoxylin Elsin, we could observe glomerular-like structures in where after staining and ulterior confocal analysis, we marker for detecting human cells in green, we detect in close vicinity with like cells in magenta, expressing podocalyx. Also, we were taking advantage of other markers as CD31, nuclear antigen, in order to further detect these endothelial cells. Collectively, our results that the CAM could act as an in vivo bioreactor, specific differentiation of kidney organoids that after an implantation of 5 days, evidence the existence of more important features of matureness of differentiation, including the presence of putative-glomerular-like capillary and mature podocyte related. And importantly, I would like to remark that these observations were never found. Organoids were not implanted in the cheek. We took advantage of kidney organoids to further perform nephrotoxic assays and also platform. So just for finishing this first part of the presentation, we took advantage of hydrogels that was mirroring the same stiffness as the cheek cam, and by developing cells derived in a soft microenvironment, we were able to further engraft these kidney organs. These are just some examples of 10 microscopy images in where you can see that the tubular structures were possessing orders here, treated as VD, and also pod-like cells that were showing important features of differentiation, podocyte-like cells of primary and secondary diaphragms that are here labeled with these red. So, as I was mentioning to you, particularly focused in the derivation of differentiation systems, including organ understanding important steps of tissue specification and differentiation. But our final goal at the end of the road is to provide cellular systems to further model human disease. So in this regard, we have recently participated in a collaboration that was led by Professor Joseph Penninger, and also in collaboration with Professor Alimi Lazimi at Karolinska Institute, provide a human platform for the study of first detection, and also to try to put in place a system for treatment. We took advantage of blood vessel and kidney organoids, early steps of SARS-CoV-2 replication in the cellular platforms, and also to test on the utility of the human recombinant soluble H2 that at that moment was already tested in phase 1 and phase 2 trials. So we want to further understand if the use of this soluble compound was able to entry into the human cells, and subsequently the viral growth also in human derived kidney organoids or blood vessel organoids. And my laboratory was in derivation of kidney for this study. So I show you an image that corresponds to kidney organoids that were derived specifically for this study. For this collaboration, we took advantage of a new protocol where kidney organoids were derived in suspension culture conditions. So these kidney organoids, we use single cell profiles in kidney organoids in order to show that are expressing ACE2, and we can detect that ACE2 is expressed in the proximal tubular-like cells and also in clusters that express also to different cell populations. So when we were completely sure that our protocol sustained for the derivation of kidney organoid cultures ascertained for the expression of other important tubular markers as SL3, and also we extract cells from kidney organoids by using Lotus Tetaglobus lectins and put culture. So collectively, this slide, what wants to represent here is that we're containing different cell types that express H2 in a similar fashion as in observing the native tissue, but also important function. So as I was mentioning to you, this work was collaboration together with our my group participated in the derivation of kidney organoids to be further infected by the group at Karolinska Institute. The group of Ali isolated SARS-CoV-2 from that was confirmed COVID-19 disease, and then after successful culture on viral of the virus, this was sequenced and further analyzed, of course, by electron microscopy, and this image shows the prototypic coronal Alimi group. So in Karolinska Institute, Lazimi performed the infections of kidney organoids that were monitored for infection with CoV-2 particles, and further assay RNA using QPs. So the progenitor of the virus was monitored by qPCR, and as expected, we can see that the kidney organoids were properly these viral particles, and also internatan of infected kidney organoids that were collected after 6 days post-infection, and we could show that the supernatants were Ibero-E6 cells. They ascertained the use of these kidney organoids as a model COVID-19 disease, so took advantage of the human recombinant soluble agent. We treat kidney organoids infected with SARS-CoV-1 or SARS-CoV-2 particles and different doses of, and we could observe that a compound truly inhibits spiral mRNA in our kidney organoids, and that this was also performed in this manner. So for the last month or 2 months, I would rather say that we are still pursuing in our collaboration. We are nowadays analyzing different conditions, of course, viral supernatant or with SARS-CoV-2 particles. This image corresponds to the organoid specimen that has been infected with a specific concentration of viral particles, analyzed 3 days post-infection, and organoids and further analyzed by confocal microscopy analysis tubular cells here in green. LTL refers to the presence of a lectin. Then we use a SARS antigen, and then we also stain the samples for H2. Our images here show that kidney cells, specifically cells of proximal tubular cell phase, SARS viral particles. This is just another representation of the same organoid, another section of the same sample in where section in which we can also ascertain the detection of SARS in LTL positive and H2 positive cells. To conclude, that our group, together with Joseph Pedinger and Ali Mirazimi, we are still working and exploring our platform, trying to understand SARS-CoV-2 host pathogen, also exploring systemic conditions that we are aware that are exacerbating or harming the progression as diabetes using kidney organoids. Of course, of bioengineering approaches to further find robust platforms for the derivation of kidney organoids and the scaling up of these platforms for further disease modeling of CRISPR-Cas9 applications for the derivation or engineered lines that are defective for H2 and SARS-CoV expression or response. And this is my last slide. I would like, of course, to thank for approaching us, for collaborating with them in this nice work that we are doing all together. Also, Ali Mirazimi and Vanessa, that was the first author of all the effort for the collaboration, of course, to move on. And this is a picture of my life to thank my funding agency. Thanks all of you for your attention, of course. Thank you so much.
Thank you very much to the presentation. It is now time for the question and answer session. To ask the speakers a question, where it says type your questions here, and then press submit. So our first question is a general question. What are the limitations of organoids? Nuria, perhaps I could direct that. Nuria, do we have you on the line? Steve, are you there on the line?
I'm on the line. So
perhaps you mention while we wait for Nuria to rejoin us in case the line, she may have dropped away.
Yes. So I think the limitations are, there is actually several layers of limitation. There is two different ways of making organoids. Either we use it from iPS derived organoid, or we actually use it from stem cells that we take directly from the different. And for the organoids which are derived from the stem cell, they are a little bit more embryonic and are not as reproducing as much the, let's say, adult organoid that we get from the stem cells, from the cells that we have in the stem cell that we will isolate from the crypt in the gut, for an example, for the intestinal organoid. So this is the main limitation from preparing the organoids. There is other limitation. I'm not sure if Nuria now is. No, probably not. The other limitation that we will have on organoid is there can be very different variability between manipulator or between labs, the way you are handling. Actually, one limitation that is very well known that a lot of people, and for an example, the lab of Nuria is working very hard in kind of standardizing, preparing kidney organ reproducible manner. And that's something many groups are actually working on.
Thank you very much. I will move on to our next question, and if and when Nuria rejoins us, perhaps she may like to add more.
Probably will.
So the next question we have is for you. It's on how organoids get infected. The question asks, infected in the first place, when the infection is on the apical side, which is obviously the organoid model. How does the virus enter the mini-gut lumen?
Yeah, this is actually a really good question. So you actually have to break the organoid open for viruses that want to enter from the apical, which is the case for SARS-CoV-2. So if you try to put this organoid, which has their closed organoid, which have their basolateral side facing outside, you will have very, very limited infection. So we are now kind of comparing the apical versus basolateral, and this is about 70%, 30%, apical basal ratio. And some viruses basically don't care; they can really infect well any side. In the kidney organoid, I am not sure that problem because it's kind of a more or less polarized phenotype, I would say. But yes, in the gut organoid, you have to consider that 2-dimension, and then you have a strict access to the apical side. And this matters very much because we have actually worked where we compare, not with SARS-CoV-2, but with other viruses, that the immune response that the cells will generate if the virus comes from the apical side or the basolateral side will actually be very different. So it's an important consideration to have. But if we want to center towards SARS-CoV-2, whether infection in the gut is initially coming from the apical, so from the basolateral side, actually we don't know where it comes from. We know it's there, but I will not speculate where it comes from.
Okay, thank you very much. Moving on to our next question. Our next question asks about microbiome, whether or not organoids also contain microbiomes or similar factors, and if that influences the research in any way.
No, so currently the organoid model that we have available in our lab, or in most of the lab, I think, in the world, is microbiome, and that's something that have to be considered. And this is definitely something we cannot ignore on the long term. How can I say? It will be impacting how the virus are responding, or how to the virus infection. The technical difficulty there is to grow cells, which could be bypassed, but you have to grow the microbiota, which is a very complex population require for the gut, at least hypoxia, and this is becoming technically challenging. This is something we are currently working on, and many other labs are doing, but I think it will take some years before we can do that.
Thank you very much. Few questions ask more about all other infectious diseases. One in particular asked about cancer. Would you be able to summarize your response to that?
Yeah. So organoids, I think for the last 5 years, used for studying infectious disease. I mean, it really started with Hans Clevers and Sina Bartfeld, bacteria infection using enteric gastric organoid. And after that, Marie Estes come up with norovirus infection. We have enterovirus with Christian Vobus. There's a lot of work. It's a really nascent field. And in terms of, cancer organoids, so now we are talking about organoids derived from the tumors. This is an active field of research doing personalized medicine from cancer organoids, like Michael Boutros here is preparing organoids from patients and screening drugs to do personalized medicine for carcinoma, colon carcinoma organoids. It's a fast evolving field where we now have so many people in so many fields.
Thank you very much. Our next question, does a cytokine storm phenomenon take place in the intestines? Interferon treatment an option for inhibiting SARS-CoV-2 in the intestine, since you show that interferon treatment inhibits SARS-CoV-2 in the system? And as a further question, how do you address the stability of the virus in the intestinal gastric juices in the model that you have in organoids?
Okay, so it's a long question. I would be direct. I believe, and I really think there is no real evidence for that yet, is that the cytokines happen in the gut. I mean, the gut is the biggest immune system in our body, but there has been no study that really looks at this, at least to my knowledge. I mean, so many papers now, but we see that the intestinal cells are highly infectable, produce a lot of pro-inflammatory response. So a cytokine storm coming from the gut is likely possible, but needs to be tested in biopsies. Now, the second part of the question is whether the stability of the virus in the gastric fluid. So I think there is a good news for us. We think that the virus is actually released in the apicals by the cells, so toward the lumen of the gut. And the virus seems to be non-stable in gastric fluid. And that's from Songyang Ding in WashU that actually demonstrated this very nicely, that as soon as you put the virus in gastric, so I will speculate that there is no much of a transmission, or very little if there is some.
Thank you very much. I would now like to question to Nuria. We have a question here asking, are the cells in the nephron-like structures induced in vitro in the organoid higher or lower levels of COVID-19 receptors?
Hi, so we did some single cell profiling to show that ACE2, for example, is expressed in the tubular-like cell. also in the native kidney. So we can induce the formation of the nephron-like structures in 3 dimensions in suspension, and we can action by single cell profiling and also by using immunostaining and immunofluorescence and whole immunostaining, as we did show in the presentation. Is what we are doing and we are looking for is for the expression of other receptors that have been also involved in the entrance of the virus, CD147, and also co-receptors of ACE2, ASTRIMS2. So it's good to see that, in our case, in 16 days, we can derivate kits that are still quite embryonic, and this can go to your first question. So still the field develop more strategies for improving the extent of differentiation, especially if we work with organoids derived from human pluripotent stem cells. But the good news is that in few days, in weeks, we can have, as Steve was mentioning in his talk, the machinery in the cells for as platforms studying COVID-19 related issues using these platforms.
Thank you very much, Nuria. And very sadly, that is where we need to finish for today. We are aware that there are a lot of questions that we managed to get to, and we will get back to you offline after this broadcast finishes using the details that you've provided. I'd like to thank both Dr. and Dr. Nuria Montserrat once again for their presentations and for answering your questions today. First, sponsor Nikon, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this at any time on demand at nature.com/webcast. Thanks for watching, and I hope you'll join us again soon.